Hey there, everyone, and a really big welcome to Mr. Martin's IB biology class.
Hello.
We know you're getting into D3.2 right now, all about inheritance, and tackling those big questions like, what patterns do we see in plants and animals?
hmm And what's actually happening at the molecular level?
Exactly. So think of this deep dive as uh, unlocking those secrets of how traits get passed along.
Yeah, we're going to explore the core ideas of inheritance, focusing on eukaryotes that reproduce sexually.
Right.
And it's really important to remember meiosis. That's the process making the haploid gametes egg and sperm. Half the chromosomes,
Okay.
they fuse, form a diploid zygote, full set of chromosomes. That's the starting point. And those diploid cells, they've got 2 copies of every autosomal gene, the non-sex chromosome genes.
And for all you IB bio students listening, getting this stuff is crucial, not just for exams, obviously, but it underlies so much biology.
Totally.
Our goal here is just to make these ideas clear, maybe even interesting. So what's the plan? What are we covering?
Okay, so we'll kick off with Gregor Mendel. His pea plant work, P, F1, F2 generations, Punnett grids the basics.
Right, the classic stuff.
Then we'll nail down the key terms: genotype, phenotype, homozygous, heterozygous, alleles, dominant, recessive, all that vocabulary.
Essential.
We'll also look at how the environment can shape traits, that's phenotypic plasticity. Then multiple alleles, like the ABO blood groups,
Yeah, good.
and codominance, incomplete dominance,
Yeah.
then shift to sex-linked inheritance. Hemophilia is a great example there.
Okay.
And finally, we'll look at pedigree analysis, how we trace these things in families.
Sounds like a solid plan. Okay, let's dive in with Gregor Mendel. His choice of pea plants was pretty smart, wasn't it?
Oh, absolutely brilliant. For a few reasons, they grow relatively quickly, short generation times.
So you see results faster.
Exactly. And, no major ethical hurdles like you'd have with animals. Plus, they produce tons of offspring.
Which means more data, better patterns.
Precisely. Large sample sizes were key for him. But it wasn't just the choice of plant. It was his method. Super careful.
How so?
Well, he actually controlled the pollination. He'd remove the anthers, the male parts, from one flower to stop self-pollination.
Uh-huh. Okay.
Then he'd manually transfer pollen from another specific plant he'd chosen. He even put little paper bags over the flowers afterwards.
To stop stray pollen getting in.
You got it. Preventing accidental crosses. This let him track traits very precisely across the generations. The P generation, the parents,
Right.
their kids, the F1 or first filial generation.
Okay.
And then when he crossed the F1s together, their offspring were the F2, second filial generation.
And that leads us to the Punnett grid. It's a tool, right? Not like a crystal ball.
Exactly. It helps us figure out the probabilities of different genetic combinations in the offspring. It doesn't guarantee a specific outcome in, say, one small family,
Yeah.
but it shows the likelihoods.
Okay, so let's do a quick example, like a simple one. A monohybrid cross.
Sure. Let's say homozygous dominant for round seeds. We can use big R for the genotype.
Okay, big R crossing with.
So how about a heterozygous plant? So
Right.
big R, little r, one dominant, one recessive allele.
Got it. So big R crossed with big R, little r. What does the Punnett grid show us?
All right. The big R parent can only make gametes with big R.
Makes sense.
The big R, little r parent makes gametes with either big R or little r. Half and half.
Okay.
So when you fill out the grid, you see the possibilities for the offspring are big R, and big R, little r.
And the ratio.
It comes out as a 50% chance of being homozygous dominant, big R, and a 50% chance of being heterozygous, big R, little r.
Okay, clear. Now, we've used the word allele a few times. Can we just pin down exactly what an allele is for everyone?
Good idea. So think of a gene as like an instruction for a trait, seed shape, for example.
Right.
Alleles are just different versions of that same gene. They have slightly different DNA-based sequences.
Ah, okay. So small DNA differences.
Yeah. One allele might code for round seeds, another slightly different sequence codes for wrinkled seeds. Same gene, different versions, different outcomes.
Gotcha. And that ties into genotype and phenotype. Genotype is the allele combo, right? Like big R, little r.
Exactly. It's the genetic makeup an individual has for that trait.
And phenotype.
That's the observable trait, which you actually see. So for both big R, and big R, little r genotypes, the phenotype is round seeds.
Because big R is dominant.
Okay.
And you mentioned homozygous and heterozygous.
Right. Homozygous means having 2 identical alleles for a gene. So big R is homozygous dominant. Little r would be homozygous recessive.
And heterozygous.
That's having 2 different alleles, like our big R, little r example.
Okay. And it's important to remember not everything we see is purely genetic, is it?
Oh, definitely not.
Hmm.
Some things are almost purely environmental, like, getting a tattoo or dyeing your hair.
Sure.
Other things, like your ABO blood type, are pretty much entirely determined by your genes. But most traits, think about height or maybe even susceptibility to some diseases, are a mix, a combination of your genes and your environment. Diet, lifestyle, exposure, lots of factors.
Right. So back to alleles. We mentioned dominant and recessive. Dominant always shows up if it's there.
Pretty much. A dominant allele expresses itself in the phenotype, whether you have one copy, heterozygous, big R, little r, or two copies, homozygous dominant, big R, big R.
And recessive.
The recessive alleles effect is only seen in the phenotype if there's no dominant allele present. So only in the homozygous recessive state, little r for wrinkled seeds.
Okay, that makes sense. Now, something related but different, phenotypic plasticity. This sounds complex. It's not about DNA changing, is it?
That's the crucial point. No, the DNA sequence, the genotype stays the same. Phenotypic plasticity is the ability of one genotype to produce different phenotypes depending on the environment.
So the same genes can lead to different outcomes.
Exactly. It's about changes in gene expression, not the genes themselves. The environment triggers certain genes to be more or less active.
Can you give us a common example?
Sure. Think about getting a tan. When your skin is exposed to more UV light from the sun,
Right.
it triggers cells to produce more melanin pigment. Your skin gets darker. That's a change in the phenotype.
But my genes for melanin haven't mutated or anything.
Nope. Your genotype is the same. The UV exposure just upregulated the expression of those melanin genes. And, importantly, it's usually reversible. Less sun, the tan fades.
That's a great example. Okay, so far we've mostly talked about genes with two alleles, like big R and little r, but it can get more complicated, right? Multiple alleles.
Definitely. While any one person only gets two alleles for a gene, one from each parent, within the whole population, there can be many, many different versions or alleles for that single gene.
Wow. Any examples?
Well, there's a gene in apples, the S gene, involved in self-incompatibility. Believe it or not, there are something like 32 known alleles for it.
32 versions. That's amazing. Explains some apple variety, I guess.
It contributes, yeah. But a more familiar human example is the ABO blood group system.
Yes. A, B, A, B. Oh, how does that work with alleles?
Okay, so there's one gene, but three main alleles in the human population, IA, IB, and I.
I, B, and little I. Got it.
IA leads to type A antigens on red blood cells. IB leads to type B antigens. And the little I alleles doesn't produce either antigen.
So what genotypes give what blood type? If you get two IA alleles.
IAI gives type A.
Right.
So does IAI because IA is dominant over I.
Okay, same for B.
Yep. High BB or IB both give type B blood. IB is also dominant over I.
What about little I?
Little I is recessive, so you only get type O blood if you have two copies.
Okay. But what if you inherit IF from one parent and IB from the other, IAIB?
Now that's where codominance comes in. Both IA and IB are dominant, but they're codominant with each other.
Meaning
Both are fully expressed. Someone with the IIIBA genotype has both A antigens and B antigens on their red blood cells. They have type AB blood.
okay, so codominance is like both showing up equally, not one masking the other.
Exactly. Not a blend, but both present.
Let's try a cross. Say a mother with type O blood.
Okay, so your genotype must be 2.
Right. And a father with type AB blood.
His genotype is IAIB.
What blood types could their children have?
All right, let's think gametes. The mother, 2, can only make eggs carrying the I allele.
Okay.
The father, IAIB, makes sperm carrying either IA or IB, roughly 50-50.
Right.
So using a Punnett grid, if an I egg meets an IA sperm, the child is IAI.
Phenotype.
Type A, because IA is dominant over I.
Perfect. If an I egg meets an IB sperm, the child is IB. Phenotype. Type B.
Same logic.
Exactly. So the children can only be type A, IAI, or type B, IB. There's a 50% chance for each.
Interesting. So none of the kids would have type O, like the mom, or type B, like the dad?
In this specific cross, nope. Shows how inheritance isn't always straightforward averaging.
Okay, so that's codominance. What about incomplete dominance? How's that different?
Incomplete dominance is different because the heterozygote phenotype is an intermediate kind of a blend between the two homozygous phenotypes.
A blend, like mixing paint. Sort of. The classic example is the 4 o'clock flower, Mirabilis jalapa. You can have plants homozygous for red flowers, say RR genotype,
Okay, red.
and plants homozygous for white flowers, WW.
White.
If you cross RR with WW, the heterozygous offspring, RW, aren't red and they aren't white. They're pink.
Ah, so the heterozygote is a mix, pink, not like type AB blood where you see both A and B features separately.
Exactly. If there were codominance in the flowers, maybe you'd see flowers with patches of red and patches of white. But with incomplete dominance, you get the intermediate pink color.
Got it. Blend versus both showing up side by side.
Okay, let's switch gears a bit. Sex determination and sex linkage. In humans, it's the X and Y chromosomes. Yeah,
That's right. Biological sex is typically determined by that 23rd pair of chromosomes. Females usually have XX. Males usually have XY.
and it's the dad's contribution that decides it.
Correct. The egg from the mother always carries an X chromosome. The sperm from the father carries either an X or a Y.
So if an X sperm fertilizes the egg,
You get XX, biologically female.
and if a Y sperm fertilizes the egg, you get XY, biologically male. Simple Punnett square shows it's roughly a 50-50 chance.
Okay. And because these X and Y chromosomes are different, genes located on them have different inheritance patterns, sex-linked genes.
Exactly. The X chromosome is much larger than the Y and carries a lot more genes. Genes found on the X are called X-linked genes.
And the Y.
The Y chromosome has relatively few genes, and for many genes on the X, there's no corresponding allele on the Y,
How do we write the alleles for these?
usually as superscripts on the X. For example, if we're talking about a gene with a dominant allele age and a recessive allele age, we'd write XH and XH.
And for the Y chromosome?
For a gene only on the X, the Y is often just written as Y, sort of indicating it doesn't carry an allele for that gene.
Okay. And a classic example of an X-linked condition is hemophilia. What is that exactly?
Hemophilia is a group of genetic disorders where the blood doesn't clot properly. It's typically caused by a recessive allele on the X chromosome.
X-linked recessive. Okay.
This allele results in a deficiency of one of the clotting factor proteins needed to form blood clots. So affected individuals can bleed for longer than usual after an injury.
And because it's X-linked, it affects males and females differently.
Yes, significantly. Let's think about females first. They have two X chromosomes.
XX.
So they can be XHXH, two normal alleles, normal clotting,
Right.
or they can be XHXH, two recessive alleles. They have hemophilia,
Okay. What's the third possibility?
XHXH, one normal, one recessive allele. These females are carriers,
Carriers.
meaning they carry the recessive allele, but they usually have normal blood clotting because the normal XH allele is dominant and masks the effect of XH.
Got it. Now, what about males? They're XY.
Right. They only have one X chromosome.
Yeah.
So they can be XHY. They have the normal allele, normal clotting.
Or
Or they can be XHY. They have the recessive allele, and since there's no second X chromosome with a dominant allele to mask it,
they have hemophilia.
exactly. Males cannot be carriers for X-linked recessive traits. They either have a condition or they don't.
That explains why these conditions are much more common in males.
It does. Let's do a quick cross. Say a normal male marries a carrier female.
Okay, so male is XHY, female is XHXH. What about their kids?
Let's see, Punnett square time again. The male produces XH sperm and Y sperm. The female produces XH eggs and XH eggs.
Okay.
Possible combinations for daughters,
Huh.
XHH, normal female, and XHHH, carrier female.
50% chance daughter is normal, 50% chance she's a carrier.
Right. Possible combinations for sons, XY, XHY, normal male,
Yeah.
and XHY male with hemophilia.
Ah, so 50% chance a son will be normal, 50% chance he'll have hemophilia.
Precisely.
Right.
So overall, in this family, there's a 25% chance of having a child with hemophilia, and that child would be male.
Okay. That really clarifies X-linked inheritance.
Hmm.
Now, how do we track these sorts of patterns or any genetic trait through actual families? Pedigree charts.
Yes, pedigree charts are the standard tool. They're essentially family trees drawn using specific symbols.
Symbols like what?
Circles represent females, squares
Yeah.
represent males. If a shape is shaded in, it means that individual is affected by the trait being studied. Unshaded means unaffected.
How are relationships shown?
A horizontal line connecting a circle and a square means they've mated or produced offspring. A vertical line dropping down from that connects to the offspring generation.
And looking at these patterns helps figure out inheritance.
Absolutely.
Yeah.
You can often deduce the mode of inheritance, like is it dominant or recessive, autosomal or sex-linked, by looking at who is affected across generations.
Can you give an example?
Sure. If you see a recessive disorder where affected individuals, shaded shapes, have unaffected parents,
Right.
you can usually infer that the parents must both be heterozygotes, carriers, for that recessive allele. The affected child inherited one recessive allele from each parent.
Ah, okay. You can work backwards.
You can. Pedigrees also visually show why conditions caused by rare recessive alleles are more common in situations of inbreeding, like marriages between close relatives.
Because relatives are more likely to share the same rare alleles.
Exactly. They might inherit the same recessive allele from a common ancestor, increasing the chance that their child gets two copies and expresses the disorder.
Makes sense. Okay, we've covered lots of specific inheritance patterns. Let's zoom out a bit. We talked about traits like blood type or hemophilia clear categories. That's discrete variation, right?
Correct. Discrete variation means traits fall into distinct, non-overlapping categories. You either have type A blood or type B, etc., or you have hemophilia, or you don't,
And these are usually controlled by.
often by a single gene or maybe just a few genes, with minimal influence from the environment.
But then there are traits like height or skin color where there's a whole range,
Yeah.
continuous variation.
Precisely. Continuous variation is where phenotypes show a smooth spectrum of values between two extremes. Think about the range of human heights in your class.
Yeah, not just tall and short.
Right. These traits are typically polygenic, influenced by many genes acting together, and they're often significantly affected by environmental factors too. Nutrition affects height, sun exposure affects skin color.
Okay. And if we have data for a continuous variable like the heights of all the students in Mr. Martin's class, how can we visualize that distribution? Box and whisker plots.
Yes, box and whisker plots or just box plots are excellent for summarizing continuous data distribution.
What do they show?
They give you a quick visual summary based on 5 key numbers. The minimum value, the
Yeah.
first quartile, Q1, the median, the third quartile, Q3, and the maximum value. Sometimes they also explicitly mark outliers.
Okay, Q1 and Q3, what are those again?
Q1, the first quartile, is the value below which 25% of the data points fall. The median is the middle value, 50th percentile.
Yeah.
Q3, the third quartile, is the value below which 75% of the data falls.
So the box part of the plot.
The box is drawn from Q1 to Q3, so it contains the middle 50% of the data. The line inside the box marks the median.
And the whiskers.
The whiskers typically extend out from the box to the minimum and maximum values in the data set, unless there are outliers.
Outliers. How do we know if something is an outlier? Is it just if it looks really far away?
There's actually a mathematical rule for it. It's not just a guess. You first calculate the interquartile range, or IQR.
IQR, yeah.
IQR is just Q3 minus Q1, the range of the middle 50% of the data.
Okay, Q3 minus Q1. Then what?
Then you calculate the fences. The upper fence is Q3 plus 1.5 times the IQR. The lower fence is Q1 minus 1.5 times the IQR.
Right.
Any data point that falls outside those fences, either above the upper fence or below the lower fence, is formally considered an outlier. The whiskers on the plot usually only extend to the highest and lowest data points within the fences.
Got it. So it's a defined calculation, not just a feeling.
Exactly. Important to remember that.
All right, Mr. Merton's IB biology class. Wow, we've covered a lot of ground in this deep dive on inheritance.
I certainly have.
From Mendel and his peas, Punnett squares, all that key terminology.
Genotype, phenotype, alleles.
Then into multiple alleles, codominants like blood types, incomplete dominants like those pink flowers.
This linkage with hemophilia, pedigree charts.
And finally, wrapping up with discrete versus continuous variation and how to visualize data with box plots, including those outliers.
Yeah, it's a foundational area of biology. And remember, understanding these principles isn't just, for the classroom.
Right.
It's key to understanding evolution, why populations have genetic diversity, how we breed better crops. It connects to so much else.
Absolutely. So to everyone in Mr. Martin's class, we really hope this session helped clarify things and maybe even sparked some more curiosity.
Keep asking questions.
Yeah. And if you found this useful for your IB studies, please do hit that like button, subscribe to the platform wherever you're listening. It really helps us know what's valuable for you. And definitely drop a comment below.
Yeah.
Any questions you still have, other biology topics you want us to tackle, let us know. Your input genuinely helps shape what we do next.
We read them.
We do. And please share this with your friends. Anyone else in the IB Bio program who might find it helpful, spread the knowledge.
Definitely.
Okay, so to leave you with something to think about.
The final thought.
Yeah. Considering everything we've discussed, especially that interplay between genes and the environment, phenotypic plasticity, how might our increasing understanding of that plasticity shape the future?
Huh.
Think about approaches to human health or maybe agriculture.
Ooh, good one.
Ah.
How can we leverage that flexibility potentially?
Yeah.
Lots to consider there. Something to chew on after the deep dive ends.